Today's class is about meiosis. So meiosis is not difficult conceptually, but there are a ton of vocab terms that are required to understand it, so it's very easy to get lost in the weeds here. I would say this is actually the most vocab heavy process of the year, but again, that overall concept of meiosis is not that difficult in terms of being able to describe it and what happens. So buckle up and hold on to your butts. Let's get going. Alright, so pretend you know nothing about cell division or anything about mitosis, which is what we've been covering. Basically, if you were just to compare this picture, picture 1, and picture 2, hopefully you'd pull out a couple of key differences. The most obvious being that there's one cell here and that there's 4 cells here. You might also notice that in this cell we have 1, 2, 3, 4, 5, 6, 7, 8 different colored lines, and in each of these cells over here there are 4 each. The third detail that you might notice, which is probably the hardest to see, is that each one of these lines is a mixture of 2 different colors. So here we have an orange and a red, here's a dark blue and a light blue, the yellow isn't mixed, and then the green and the light green. But essentially what this means is that each one of these 4 centers here are different from each other, whereas here all of the colors are uniform and the same. So that's kind of the overall, if you can see these differences, you can understand the concept of meiosis, because that's all there really is to it. This is our initial cell before meiosis divides it, and then afterwards you'll end up with 4 unique cells. The terminology in explaining how you get from picture 1 to picture 2 is what's difficult. That's what this is going to be. This is what this lesson is going to be about. So what is produced by meiosis? Being specific in terms of the number, type, and genetics of the cells being produced, we could say that meiosis produces 4 haploid, genetically unique gamete cells. So right now we may not be sure what this term actually means, but by the end of this lecture, hopefully you will have an understanding. So if nothing else, know this about meiosis. It occurs in germ cells. These are your reproductive cells, and the germ cells undergo this very special type of division to become something called a gamete. If you're a female, that's an egg. If you're a male, that's a sperm. The parent cell, the germ cell, goes through 2 rounds of division as opposed to mitosis where there's just 1 round of division, and it becomes 4 daughter cells. Again, these cells are called gametes. Each gamete is going to be genetically unique with only half the amount of chromosomes. That's important. And then gametes combine to make new life. That initial new cell that's created is called a zygote. So when one gamete combines with another, an egg and a sperm, they create that first new cell, which is a zygote. Note that these notes, this right here, tells you what meiosis does, but not how it does it. So this is important to know, but the details, again, are going to be forthcoming. Alright, so a couple of key terms just to get out there because we need to have these as part of our discussion of meiosis. First is haploid. A haploid cell is a way to describe a cell that has half the number of needed chromosomes in order to function. So all different species have different numbers of chromosomes. Humans have 46, bananas have something like 94, tomatoes have something like 1,000, I'm not too sure. But all these are going to be even numbers because the number of cells needed, or the number of chromosomes needed to function can be cut in half by the process of meiosis. And so when you're referring to those cells with half the number of chromosomes, those are haploid cells. Conversely, a cell with a full set of chromosomes is called diploid. A gene you already know, but just to refresh you, it's a segment of DNA that codes for a trait. Specifically, it codes for instructions on how to build a protein, which then manifests itself as something called a trait or a characteristic. But the new twist to that is something called an allele. An allele is an alternate version of the same gene. Your DNA carries 2 alleles for every gene. So let me try to illustrate the differences between gene and allele. A gene is something like hair color or eye color. It's a category, whereas an allele is the actual adjective used to describe that category. So if your hair color gene is brunette, that's referring to your allele. So alleles might be brunette, blonde, redhead, dirty blonde, something like that. A gene for eye color would simply code for your eye color, or that's the category, but the alleles can be the different colors, blue, brown, hazel, green, that kind of thing. When people say you have really good genes, that's not really all that accurate. What they really mean is you have really good alleles, because everybody has the same genes. We all have the gene for hair color. We all have genes for eye color. We all have genes for 2 arms and 5 toes and 5 fingers, or sorry, 10 toes, 10 fingers. But what's different are our alleles. Sometimes we do have the same alleles as well across the species, but that's what gives us our variety, is the alleles that exist, which are variations of a gene. When you get genes, you inherit them in pairs. One of the pair comes from your mom and one comes from your dad. Humans happen to have 23 pairs of chromosomes for a total of 46 chromosomes in every single cell. So you'll notice that chromosomes are, on this picture over here, this is a full set of a human set of chromosomes. They're organized by size. So the tallest or the biggest chromosomes with the most genetic information on them is usually the first chromosome in a set. Then the second biggest would be the second in the set, and so on and so forth, all the way down to the second to last, where that's the smallest one. The last set of chromosomes are reserved for something that will help determine gender, but we'll talk about those later. So when you look at a pair of chromosomes, like this pair of number 5 chromosomes, if you were to zoom in on them, what you would notice is that each segment is going to categorize or code for a gene. So here's an A gene. We don't know what that codes for. Maybe that's the gene for hair color. Maybe the B gene is the gene for your ability to digest milk. But sometimes if you compare the gene that you get, or the chromosome that you get from your mom compared to the chromosome that you get from your dad, they're different. Sometimes they're the same. So notice here you have a big A, here's a little a, here's a little b, and then on the chromosome from dad is a big B. But in this situation, you happen to inherit both a big C from mom and a big C from dad. So whatever genes and alleles these happen to be, they're the same. So there's no way but to exhibit whatever trait this happens to be. This is important. These are not sister chromatids. So we've learned that sister chromatids are identical copies of a chromosome that are made during DNA replication, but don't confuse these with sister chromatids. They're not connected with a centromere or anything like that. These are what you would call homologous pairs because they're similar, but they have some key differences. Now, understanding the difference between sister chromatids and homologous pairs is very, very crucial to understanding meiosis. Sister chromatids have identical genes and alleles. They're counted as one chromosome. They were made during S phase because one of the chromatids virtually or exactly copied itself and then paired up with itself. Homologous chromosomes are different. They have identical genes, but the alleles don't have to be the same. Sometimes they are, sometimes they're not. They're counted as a pair of chromosomes, so they're counted individually. One of that pair came from your mom, and the other one came from your dad. You inherited homologous chromosomes, but you make sister chromatids. Again, here's another kind of illustration of that difference. Here's a sister chromatid. This one was the original, and then this one is a clone of it. Notice that what I'm trying to illustrate with these colors is that they're the same across the board, right? So 2 copies of the same book does not equal 2 different essays. It's the same genetic story, so it counts as 1 chromosome. Here we have a homologous pair. We have the same genes, whatever those genes happen to code for. It's the same category, but there might be difference in how that code is presented. So this might be the gene for your nose shape, and this one is a big nose, and this one would be a medium-sized nose or something along those lines. It's kind of like 2 versions of the same story, so this would be considered 2 different stories. When they come together, because they do come together at some point in meiosis, it's referred to as a tetrad. It's a pair of homologous chromosomes. Here's the sister chromatid from mom, and here's the sister chromatid from dad. All right, keeping track of chromosomes and chromatids is kind of tricky, so I'll go over it really quickly right here. Here we have, let's say this is a single chromosome organism. So this organism has 1 chromosome in it. Here it's 1 chromosome, and it counts as 1 chromatid. During S phase, when this chromatid copies itself, you will now have 2 chromatids that are linked together by a centromere. One is on this side, and one is on this side. But because they're identical, they're still counted as 1 chromosome. During mitosis, when these cells split, one chromatid is going to go this way, and the other chromatid is going to go that way. Because they are now in different locations, this counts as a chromosome, and this counts as a chromosome, whereas before they counted as 1 chromosome together. So it's kind of a tricky numbers game, but you'll get the hang of it. All right, so once again, this is what would happen in the beginning of mitosis and meiosis. Here would be your normal set of chromosomes, all 23 pairs, and then during S phase, each one of these chromatids is going to copy itself and link up to itself to create a sister chromatid. That would be right there. So this always happens in preparation for division, whether it's mitosis or meiosis. In mitosis, the division separates just the sister chromatids and then you're done. But in meiosis, there's actually 2 rounds of division. The first is going to pull the homologous pairs apart, and the second will pull the sister chromatids apart. I'll show you exactly how that looks. So a quick vocab recap, because at this point you will need to have a firm understanding of these 8 terms in order to have a discussion about how meiosis goes forward. So make sure you have these notes, make sure those notes are taken down well and that you can refer back to them. You will definitely need to know exactly what these terms are as we go through them and as they are part of our discussion. Alright, so the cell cycle of a germ cell is different from the cell cycle of a somatic cell. They're similar, but a germ cell is going to undergo meiosis instead of mitosis. And the key difference there is that meiosis has 2 rounds of division, whereas mitosis only has 1. So in M phase, you'll go through this first round of meiosis where the homologous chromosomes are pulled apart, and then in meiosis 2, you'll go through a second round of division where the sister chromatids are pulled apart. Let's look at that in more detail. So in interphase, just as in cells that undergo mitosis, during interphase the chromosomes are going to duplicate and create sister chromatids, which again are identical copies. During what subphase do chromosomes duplicate? I hope you guys know that. That would be S phase. Alright, in our cells, in human cells, we have 46 chromosomes, but in the model that I'm going to be using, there's going to be 8, for example, because that would be more simplistic to follow. It's too difficult to keep track of 46 chromosomes. So here are our 8 chromosomes. They're homologous pairs, one from mom, one from dad, mom, dad, mom, dad, mom, dad. Now, during S phase, each one of these chromatids is going to copy and create a sister chromatid of itself. So again, this is a homologous pair of sister chromatids. All right, let's keep going. So what you can do if you want is you can grab some of these linking cubes and you can do this model along with me, or you can just look at this picture and see what's going on. If you were to build a karyotype, which is a complete set of chromosomes for an organism, let's say we have 8 chromatids for this individual. The mom's chromosomes are going to be this blue block, this yellow one, this red, and this green one, and the chromosomes that this individual inherited from dad would be the purple, white, yellow, and brown. This represents a complete set of chromosomes. This is known as diploid, so it's a full set of chromosomes. How many chromatids are there? Well, there's 8, and how many chromosomes? There's 8 as well because they're individual copies. Individual genetic differences between them. They're not identical. So what we're going to do first, before we start dividing, is we're going to make a copy of each chromatid and attach it using a black block. So you can see the black blocks in the middle here. Those will be centromeres. When does copying occur? S phase. What are the copies called? Sister chromatids. What's the black thing? That's a centromere. And how many chromatids are there? Well, now those have doubled. So there are 16 chromatids now. But because they're just copies, they only count as 1 chromosome. So there's still just 8 chromosomes. Quick review before we show the whole process. Right here we have chromosomes that are different from each other. So we'll call this a homologous pair of chromosomes. Here we have chromosomes that are duplicated and they're exactly the same. So this pair and this pair are identical. So we'll call those sister chromatids. But when you compare this one to that one, that's homologous. So together, this would be called a tetrad. All right, let's begin. So in meiosis 1, prophase 1, the sister chromatids are attached and the homologous chromosomes will begin to find each other. So these are literally going to start moving around the cell and start linking up and getting close to each other like this. They'll just start meeting up towards each other and finding their pairs in the cell. Homologous pairs and sister chromatids are perfectly matched up at some point, and the resulting structure, again, is called a tetrad. Next, crossing over is going to occur. This is extremely important. This is the exchange of genetic material between homologous chromosomes. So take a look down at this cell right here. You'll notice that before these both were solid blue because they are identical copies. They were sister chromatids. But what happens during crossing over is little bits of genetic information literally swap places and they exchange to be on different chromosomes. So that happens with each one of our chromosomes here. A little bit from this one goes to this guy, and a little bit from this one goes to this guy, and they all exchange bits and pieces of genetic information. If you wanted to show that on our model, what you can do is you can exchange a block from each homologous pair so that the sister chromatids are no longer identical. This exchange is called crossing over, and what's it do? Well, it mixes DNA from mom and dad. This is going to be really important if you're trying to make children that are genetically different from yourself. All right, crossing over, again, hugely important. The alleles from homologous chromosomes trade places. Take a minute, write down that note. It's super, super important that you have this understanding of what crossing over does. The resulting chromosomes are not really sister chromatids anymore because they're not identical. So instead we call them, or we refer to them as recombinant chromosomes. So here is our original tetrad. Here's a sister chromatid right there. Here's a sister chromatid. The red and green make a homologous pair. They literally cross over and then swap genetic information, and these are now referred to as recombinant chromosomes because they've been recombined. All right, our next phase in meiosis 1 is metaphase 1. Here, the recombined chromosomes are now aligned in the center. The random way that they line up is called independent assortment, and this shuffles the genes from mom and dad. So this gene could have been on this side, and this one could have been on that side. So let's say the darker colors are from mom and the lighter colors are from dad. It could have been like that, or it could have been like this. Same thing down here. Dark could have been on this side, lighter could have been on that side. Oops. And all of these randomly line up so that different chromosomes from your mom and dad are on different sides of what's called the cell equator, right down the middle there. That's also important because when the cell divides, it's going to pull these chromosomes apart, and in each time when it pulls apart, it's going to contain a mixture of mom and dad's chromosomes. So again, with our model instruction, you can randomly line up maternal and paternal homologous pairs in the center of the table. This process is called independent assortment, and it too is a further mixing of mom and dad's DNA. So you can see this chromosome, which is recombined, is mostly from mom, but a little bit from dad down there. This again is mostly mom, but a little bit of dad's genetic material. Here it's mostly dad and a little bit of mom's material, and mostly dad and a little bit of mom's material. So there's this mixing of genetic material here and there. All right, in anaphase 1, the homologous pairs are going to separate. So the sister chromatids are still tied together. Notice that centromere still exists there, but what's happening is the homologous pairs are getting pulled apart to different sides of the cell. Note, the sister chromatids aren't really identical anymore because of crossing over. Independent assortment, which is what's happening here, ensures that each gamete, the egg or sperm, produced will contain a mixture of your mom and dad's alleles so that your children are a combination of their grandparents. So again, to go with our model, you're going to separate the homologous pairs but keep the sister chromatids together. Are the linked chromosomes still sister chromatids? Well, not really, because they're no longer identical. They're referred to as recombinant DNA. All right, our last phase in meiosis 1 is telophase 1. Here, cytokinesis creates two daughter cells, each with a mixture of sister recombined chromatids. So you can begin to see how this genetic diversity is shaping up. This cell is not going to be identical to this cell whatsoever because of the exchange of material and because you've randomly aligned the mother's and father's DNA. All right, let's take a break and talk about meiosis 1. How many cells are there after meiosis 1? 2. Are the number of chromosomes haploid or diploid? Well, this question is kind of tricky to answer. It depends on how you count a chromosome. If you count sister chromatids as one chromosome, then it's haploid. But really, they're not sister chromatids anymore. And so you can make the argument that because they're unique, you can count them as two chromosomes. And if you do, then you would consider the cell to be diploid at this point. What major events happen during this process? This is big. This is crossing over and independent assortment. They ensure that each gamete produced will be a genetically unique combination of your mother's and father's genes. if I can stress one aspect of the lecture so far, it's crossing over an independent assortment. Really, really important. Time out for the bigger picture. You've seen this slide before. Where we're going in this unit is evolution, and the basic fundamental tenet of evolution is genetic diversity. The more diversity a species has, the more tools, quote unquote tools, it has to overcome threats of extinction. The more diversity a species has, the more likely it is to adapt. There are 4 ways that biology encourages genetic diversity. We've already learned about one of them. It's mutations. It's a random change in your nucleotides. Some are good, some are bad. But if that mutation exists, there are other ways to mix up your DNA. One of them is crossing over, and one of them is independent assortment. We're not yet onto the 4th one, so you'll have to wait to get that one later on. Meiosis 2, alright, so basically what's going to happen here is something very similar to mitosis. Both new daughter cells begin with what is essentially the same thing as mitosis. Metaphase 2, this is the quote-unquote sister chromatids, which are really recombined chromatids. They're going to move to the middle. In anaphase 2, the sister chromatids are pulled apart. You can do that with your model if you're following along. And you can also notice if any of the groups are identical. So you'll see that this chromosome is identical to this one, but that's where the similarities end. This one's different from that, this one's different from that, and this one's different from that. So there's been some crossing over that's going to change things around. In telophase 2, the nuclear membrane returns and cytokinesis begins. So cytokinesis is the splitting of the cell. Here's the nuclear membrane returning to protect that DNA inside. And let's do like a before and after. Here's our original germ cell. What do you notice about these gamete nuclei, these 4 down here, compared to the original cell? Well, right off the bat, they have half the number of chromosomes. Up here we have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes, but in each one of these daughter nuclei, we only have 4 chromosomes. The other thing you might notice is that if you compare each nuclei to each other, they're all genetically unique. They're totally different from each other, which is a good thing if you're trying to encourage genetic diversity. So again, if you compare your before model to your after model, you can see that there's 8 chromosomes with 16 chromatids in one cell. And here you have 4 chromosomes made up of 4 chromatids in each of the 4 separate cells. All right, so one more summary. Meiosis 1 is when crossing over an independent assortment mixes your parental DNA. The homologous chromosomes are pulled apart. In Meiosis 2, the sister chromatids are pulled apart and you end up with 4 genetically unique haploid gametes. And that's it for meiosis. It's a bit of a whirlwind, but if you took good notes and you kind of get this, then I think you'll be in good shape. We'll spend some more time doing this. Thanks for listening, and let me know if you have any questions.